Welcome to Study Loop.
Hello everyone, welcome to the last question of chapter 23, problem 78. This problem tells us that when we have this compound, in order to isolate this compound, we have to use base extraction. However, if we use too much base, we will obtain an optically inactive product. Explain this drawing the stepwise mechanism and explain why this compound, regardless of how much base we use or not, we will always obtain an optically pure product. So let's work through why this is. So an optical product means that it has at least one chiral center. And as we see here, both of these have a chiral center here and here. So that's why they are optically active. But if we were to do a base extraction, we see that this proton is an acidic proton. It's alpha to this carbonyl group or this ester group here. So this is actually what we call an acidic proton. So this base can rip off this proton. So if I were to draw that, we see that, and then this is the rest of the molecule. We see that the base can very easily rip off this acidic proton here, making a bond. You make a bond, you break a bond. So these two electrons are going to go onto the carbon nucleus. So now we have those two electrons on the carbon nucleus. And there's the rest of the molecule off that oxygen here. But now this carbanion, it doesn't want to stay localized on this carbon. It would rather be neutralized. So one thing it can do is that it can actually form a new pi bond here, making a bond. You make a bond, you're going to kick off a bond, and that's going to be a hydroxide ion. And hydroxide is actually a very strong base. So you're just going to reform a base in this reaction, which is actually something very favorable that can likely happen to form an alkene bond here with the rest of the molecule. And since no other chiral centers exist in this molecule, you lost your chirality at this center. So because chirality has been lost at this center, this product is no longer optically active, and therefore you lost your optically active drug. So in order to prevent that, you need to use a very minimal amount or very mild base such that it does not rip off this acidic proton. It just separates the different enantiomers from each other. If you use too much, then it will rip off this acidic alpha proton to form an alkene elimination product. As you can see here, this is what we call, this would be a E2 elimination, you could say, because everything is happening in one step. However, for this product here, because your leaving group, so the leaving group in this case is hydroxide. So your leaving group has to be one carbon away from your hydrogen that you're ripping off. So the hydrogen we're ripping off was on the bottom carbon and your leaving group is on the carbon on top. But on this example, your hydrogen and your leaving group are all attached on the same carbon. So it is not possible to form an alkene double bond here. Therefore, since your leaving group and hydrogen are on the same carbon, no elimination reaction can occur. Therefore, regardless of the amount of base you use, you will always have an optically active product purified.